right, well, welcome everyone. Welcome to you in person there at Penn and then online. This is our first seminar of the year for our future leaders in mechanobiology series. We started this about a year ago, and this is really it's a monthly seminar giving up and coming leaders in mechanobiology. These are maybe senior PhD students or postdocs, an opportunity to network and introduce themselves to the field and talk about some of their research efforts. And they come from a variety of fields and diverse backgrounds and institutions. And so Ryan, no pressure, this is you're kicking off the year, the first all right. So let me introduce Ryan. Ryan comes from Penn. Ryan Daniels is a graduate student at the University of Pennsylvania and Dr. Rob Mauch's lab. He did his undergraduate at Cal State Fullerton in cell biology. They are a Howard Hughes Medical Institute Gilliam Fellow for Advanced Study, and the focus of their thesis is elucidating the role of mechanobiology on SOX9 and in chondrogenic differentiation. So you're going to hear a lot more about this. And Ryan, please take it away.
Okay, thanks for the introduction, Trina. And thanks for everyone for being here and for allowing me to speak today. So what I'm going to be talking a little bit about today is about some of our more recent work on, again, like Trina said, elucidating the role of mechanobiology on chondrogenic differentiation, particularly on SOX9 that I'll get into in just a bit. So there's a lot of different types of mechanotransduction as we hear, but my focus is on cytoskeletal nuclear mechanotransduction. So I'm just going to be going over a brief sort of overview of how that process works, where a cell within or on an extracellular matrix will feel a force that will promote the formation or more formation of focal adhesions, that will promote Rho to rock cofilin, that will activate actin stress fibers, and without including any of the other intermediates there. And then how the extracellular matrix will mechanically couple to the intracellular environment, particularly the nucleus, is through the link complex, where whenever a cell feels a force outside the cell, that nucleus can feel that force as well through that link complex, such as a stretch. And other groups have shown that basically when they feel this stretch, certain mechanoactive transcription factors can go into the nucleus and alter gene expression, and this can influence cellular differentiation and homeostasis. And so what I started to do was look at, okay, well, what are some different cell types that we work with in our lab where we can perturb this system to query questions on the impacts of this process on differentiation or homeostasis. And one of those cells that we're interested in is articular chondrocytes, where in native healthy articular cartilage, the cells are really round. Their cytoskeleton has been described as the actin in particular being very punctate or cortical in nature. And this will allow for certain chondrogenic markers such as SOX9, which is the master regulator of chondrogenesis, and COL2 and aggrecan, which are SOX9 target genes. However, in diseased articular cartilage, or that's been damaged, what's been described in the literature is that those cells that come in to either fill in the defect or the native cells that try to differentiate to repair the tissue will sort of form this more fibrocartilaginous-like cell type, where that's been described as where the actin changes from a cortical nature or punctate-like nature around the plasma membrane to these strong actin stress fibers that can really engage with the nucleus. And this leads to, or at least is correlated with a decrease in chondrogenic gene expression, as I've described previously. However, more what's been described, so that's only so much been like noted in vivo, most of which of this mechanism has been described in vitro, where what we find is that whenever we have active Rho or actin stress fibers binding to the nucleus, we get an inhibition of those chondrogenic markers. And so something in the literature has also described as this having either a direct impact on SOX9 activity, so it is unable to sort of do its thing and go into the nucleus to affect its gene expression. However, none of these proteins, Rho, ROCK, or actin stress fibers, as far as I'm aware, have any ability to actually go into the nucleus to directly impact gene expression. And so that was when I started thinking of what are some potential transcription factors or other proteins that could be downstream of Rho, ROCK inhibition that could actually go and have DNA binding activity to inhibit those genes. And so, or have an ability to directly inhibit SOX9. Conversely, what's been described in literature as well quite extensively is that any inhibition of Rho, ROCK, or actin stress fibers can rescue any sort of dedifferentiated chondrocyte or articular-like chondrocyte to resume those cartilaginous markers such as COL2A1 and aggrecan. And so again, what is maybe a potential transcription factor that could be affected by this? And I think sort of what I've been teasing at leading into as a broad overview is basically what's a transcription factor that's very active when Rho or actin stress fibers are present in a high mechanoactive state, and is not very active when the opposite is true. And the main candidate for this was YAP. And so that's why what we want to start to look at is whether or not YAP has any sort of suppressive chondrogenic role in this in a mechanobiologic manner. And so that led to our hypothesis that Rho mediates chondrogenic gene expression through YAP and TAZ, where YAP and TAZ is acting as a transcriptional co-repressor. And so to start, I just want to introduce our cell model. So we chose to use ATDC5 cells. This is a common chondroprogenitor cell line. And most of the seminal studies that I described in the previous slide used ATDC5 cells to really establish that model and signaling pathway. So I thought this would be a great place to build upon that. To again start us off, just to get our model set up so that we can recapitulate or reproduce data that's been seen in the past, we chose to stimulate or activate Rho further by using LPA. This is a common Rho-A agonist. And then to inhibit Rho activity and actin stress fiber formation, we chose to use a small molecule CTO4. That's a cell permeable C3 transferase that can directly inhibit a broad range of Rho-A, B, and C activity. And so what we want to do is do what most people have shown in the literature, where when we inhibit Rho using CTO4, we get an increase of chondrogenic markers, so SOX9, and a trend towards a decrease with LPA, but both a significant increase in COL2A1 and aggrecan with Rho inhibition, and a significant decrease with LPA. I want to go a little bit further to make sure that the qPCR results were not just at the mRNA level, but at the protein. And so what I chose to do was do a Western for SOX9 and found a slight but significant increase in SOX9 protein levels when Rho has been inhibited. So I thought this is great. Our model is sort of set up to really query these things. So I thought, okay, let's delve a little bit further into what can be going on downstream of the Rho pathway. But first, what we want to do, again, is most of the seminal studies that have been described really used SOX9, COL2, and aggrecan as their main readouts for assessing differences in chondrogenic differentiation. And so we thought, let's broaden that horizon and do RNA-seq to get a broad transcriptomic view of what's happening. And in here is a volcano plot of our significantly differentially expressed genes in purple. And some of the ones that I've highlighted on the sides here or within here have not been described in literature as I can tell, but show significant upregulation in genes that are very important for articular cartilage development, such as GDF5 and NKX3-2, as well as other range of chondrogenic extracellular matrix proteins that are important for homeostasis. And when we do gene ontology, one of the things that sticks out is positive regulation of chondrocyte differentiation. So I thought that was great. Conversely, when we looked at the transcriptomic profile using LPA, what we found was a broad increase in matrix catabolism genes, such as matrix metalloproteinases or MMPs. And when we do gene ontology, most of the processes that stick out are, again, things that will degrade the extracellular matrix or are found in other pathways that are found in disease, or articular cartilage disease. And so again, getting a little bit further into it, we want to look at, again, what's happening with the cytoskeleton when we perturb Rho. So what we did was we put these cells on, seeded them on glass and stained for their filamentous actin, that's in gray, their DAPI is in purple, and what we can see as expected on a superphysiologic substrate like glass, the actin stress fibers are very abundant. When we do Rho inhibition with CTO4, we can see that they're very rounded. There aren't that many stress fibers present. And with LPA, what we find again, this is on glass, so we can't really make them even for as much more than possible on glass, but we do find, qualitatively that there is an increase in those like big actin stress fiber bundles. Additionally, we want to look at, so we did a Western for YAP and TAZ and found that there was a significant decrease when we did Rho, when we inhibited Rho, as well as a trend towards a decrease with TAZ. So again, this prompted us to really think like, okay, maybe there's something going on here with YAP, but it's just more correlative at this point. And so what we decided to do to really get at that was we did a transient transfection of these cells using siRNA to knock down both YAP and TAZ. And what we are expecting is when we inhibit YAP and TAZ, is perhaps also those actin stress fibers will decrease. The cell might shrink a little bit in size. And we'll see what we get with that. And so what we found was that we did single knockouts first using both siRNA to YAP or TAZ, and then we also did a double knockdown of both siRNA to YAP and TAZ. And what we found was there seems to be show a little bit of redundancy between the two, where we're getting a trend towards an increase in all of our chondrogenic markers that we chose to assess. But we really get that really significant jump in gene expression when we both knock out both YAP and TAZ. And I have to shout out Joel Burkle here, because I was just doing YAP knockdown at first, and I was really not very impressed with the results. And then he told me you really have to knock down both. So I really thank you for that. And then what I also wanted to do, again, just at the protein level, see if that increase in SOX9 is happening at the protein level. I show here an siRNA to SOX9 to verify like our antibody is good and that we're finding a decrease with that. And we do find a significant increase with SOX9 protein levels when we knock down both YAP and TAZ. And again, just to verify that our YAP and TAZ knockdown is not being due to the loss of just mRNA YAP TAZ, but the protein, I queried whether or not this was getting significantly reduced. And what we find is that both YAP and TAZ are getting reduced when I do a double knockdown for that. I thought that was great. And now I want to assess how the cytoskeleton looks, particularly the actin. And so we seeded these cells after a few days after transfection onto glass. And as we can see here, those actin stress fibers are present. YAP is pretty nuclear across the board. But when we do our double knockdown, we can sort of see that those actin stress fibers are not as present, and those cells are quite small relative to our negative control scramble. And again, we just saying for YAP as another readout that our protein is getting really reduced here, and it's pretty dim. So we thought, okay, the protein is getting reduced. These effects seem to be pretty good. But then the next question is Rho acting through YAP and TAZ to inhibit chondrogenic gene expression? And to start to tease out this pathway, what I chose to do was, again, knock down YAP and TAZ, and then use LPA to sort of activate Rho. And basically, if what I'm thinking is that it goes Rho, ROC, actin, YAP, and TAZ, and then it goes in there to inhibit chondrogenic gene expression, if we promote Rho, but we sort of block that downstream effector, we should see basically no effect or none of those inhibitory effects that I showed previously. And if it's not, and it's acting through some other protein that's unknown, I would expect it to be able to that transcription factor or whatever protein it is to then inhibit that gene expression like I've showed previously. And so how I chose to do this was I transfected these cells, and then at day 3, I added the LPA, and I chose to do day 3 post-transfection because that was when we found that was the peak reduction of mRNA after transfection with these cells, and collected these cells for qPCR gene expression analysis at day 5, because that was where we found that, again, it's transient. So that was where we found that after the time point, we would get that start to get that increase of, or that rebound of gene expression again. And what I'm showing here is just call 2, but what we find is again that significant increase in gene expression with call 2A1 with just YAP and TAZ. And again, it's still a significant increase with YAP and TAZ and LPA, but no significant difference between, both YAP TAZ knockdowns with or without LPA. So this really, supported that, okay, we really think Rho is activating YAP to go do its inhibitory role on chondrogenic gene expression. But now what I wanted to look at was whether or not this was actually due to YAP localizing to the nucleus, or if it was just in the cytoplasm, because that's sort of an assumption I've been putting on everyone, is that it's actually going into the nucleus to do this job, but I haven't quite shown you any evidence to support that. And so what the two sort of questions that it could be doing this effect is either A, YAP and TAZ is directly inhibiting SOX9 to go into the nucleus, or it's going into the nucleus and either kicking off SOX9 or competing with SOX9 for binding of those loci to inhibit that gene expression. And so how I chose to do that was, again, to use a system where that's not glass, where again, I think most of us can appreciate that on glass, it's very stiff, and YAP is generally always very nuclear on that. And so with this, so we chose to use a system where we can modulate the substrate stiffness, so we can get a dynamic range of either pushing or pulling YAP out of the nucleus, or pushing and pulling those actin stress fibers to form or not. And so what's been shown is that varying substrate modulus can impact cell shape, actin stress fiber formation, YAP nuclear localization, and differentiation. And so what we did was we took ATDC5 cells and seeded them on a few different substrates, but what we chose were just 3 in the end, which is what I'm going to show, on fibronectin-coated polyacrylamide gels, which is a 2D hydrogel system that's just been very well established to query YAP dynamics into the nucleus. And so what I'm showing here is cells seeded on glass for filamentous actin in gray, DAPI in magenta. And as we decrease that modulus, what we're finding is that those cells get a lot smaller as expected, and those actin stress fibers are not as abundant. Additionally, what we found was that YAP, where the nuclei are outlined in white, that the nuclear localization of that gets a lot to cytoplasmic ratio, gets a lot smaller. So what we're finding is that YAP is, again, relative to the cytoplasm, a lot less nuclear. And what we found with that when we look at chondrogenic gene expression, again, because I just don't want to keep showing the same 3 genes over and over again, what we find is a stepwise increase in chondrogenic gene expression read out by COL2A1 expression, where what we find is sort of compared to glass, 55 kilopascal gels are sort of an intermediate stiff modulus in between a 5 kilopascal gel, where that's where we get that significant increase in gene expression. And so what I wanted to do again was look at, okay, well, what happens, on the system, what would happen if I were to, again, stimulate or activate Rho using LPA or inhibit Rho using CTO4 on both YAP nuclear localization and chondrogenic gene expression. And so what I did was I put cells again on a 55 kilopascal gel. Shown here is F-actin in gray, magenta and DAPI in magenta, and we see sort of again like some actin stress fibers, but not really too organized. And again, YAP is sort of a lot less. It's like there's a lot more in the cytoplasm than the nucleus compared to things like glass. And then when I use CTO4, that Rho inhibitor, almost all of the actin stress fibers are completely gone. I promise there's a cell there. And as I show here where the nuclei are outlined in white, it's almost a complete absence of YAP from the nucleus. And with LPA, when I put these cells on this sort of microenvironment and promote Rho using LPA, the actin stress fibers get a little bit more organized here, and we get a massive shuttling of YAP into the nucleus. It's almost as if these cells were on glass. And so it's quantified here where there's a significant decrease in YAP nuclear localization versus LPA where we get a significant increase And this is also read out, again, showing Col2A was as an example, but the same trend was true for other markers such as SOX9 and aggrecan, where we promote, where we inhibit Rho, and YAP is not in the nucleus, we get that increase in gene expression. And when LPA, where YAP is very localized to the nucleus, we get a significant repression of Col2A1 gene expression. And to start teasing this out again, more and more, what I chose to do is sort of combine everything where I'm doing siRNA to YAP and TAZ, and then promoting Rho using LPA, but on a polyacrylamide gel like 55 kilopascal environment. And what I find was very similar to what I found on glass, where if I inhibit, if I knock down YAP and TAZ, I'll get a significant increase in chondrogenic gene expression on a 55 kilopascal substrate. And when I add LPA, I still get that significant increase and no significant difference between those two groups. So a lot of this sort of is really, again, the start of teasing out this pathway where YAP and TAZ is negatively regulating chondrogenic gene expression. And it seems from the data that I hope to have shown you is really regulating, Rho is really regulating this through the nuclear localization of YAP and presumably its co-regulator TAZ. And so something again that what we're currently doing right now in the lab is we're trying to confirm this sort of hypothesis, or sort of really help support this hypothesis of YAP acting as a transcriptional co-repressor, whether or not it's either, again, directly binding to the SOX9 loci in the nucleus, or if it's binding to SOX9 target genes. And so using various assays like ChIP-seq. And so what I want to try and see is, okay, can almost like SOX9 has been described as like this master regulator of chondrogenesis, but can YAP or TAZ kick off SOX9 from its already bound state and then to repress that gene, to repress its target gene expression? And that's something that we're currently in the works of doing right now. We sent some stuff off to really start this assay up. It's really sort of cool. Additionally, what I'm very interested in is investigating how chondrogenic loci change with mechanobiologic perturbations using oligopane DNA fish technologies that, again, we're starting to do now. Where sort of thought is, like maybe if we inhibit Rho or put these cells on softer substrates, do these chondrogenic loci sort of come together to sort of form these sort of like transcriptionally active domains or some form of territory that can either make the binding of SOX9 to those target genes more efficient and sterically hinder repression, or vice versa. And so this is something, again, that we're really interested in, we're doing right now. And I think is really really cool. And so with that, I would really like to acknowledge the mock lab in its entirety, and of course, all the funding sources from Penn, CE, MB, and of course, HHMI. And with that, I'd be more than happy to take any questions.
Great. Very nice. All right. There were some questions I posted up in the chat, and then I guess if there are questions there in person, maybe we'll take a couple from the chat because those posted up first. And I guess if you're online, why don't you go ahead and say it? Vivek, I think you're the first.
So it's a great talk. So I had a question about the cell volume. Mm-hmm. Regulating cell volume really mm-hmm. requires, charge transport, right? Mm-hmm. The osmotic state that really decides volume. So what's the mechanism for moving water in and out? Mm-hmm. These two conditions, stiff, soft, perhaps. Mm-hmm. And is that the action of mechanosensitive channels? So any thoughts on that? That's sort of the first question.
Yeah. So the question is whether or not I have any data or any knowledge of changes in cell volume, impacting chondrogenic differentiation, or if that's perhaps a different or another mechanism as to how this could be happening. Correct.
You had this data that says cell volume is different, right? In a couple of your slides earlier.
Oh, I don't know. I might have misspoke, but I don't believe I don't have any data to support that cell volume is changing, more so that just cell I might have misspoke and used the wrong term size, but what I meant to say was more like spread area was decreasing.
Oh, I see. I got it. It's
Yeah,
not
so
the volume.
sorry,
Yeah,
I
okay,
misspoke in that.
yeah.
But
I would be very surprised if the volume changes by a lot. Okay, great.
yeah,
So
and so
yeah.
it is an important question because I think even a group here at Penn with Pacifici and others over at WashU St. Louis, particularly Gilliam, have really described of osmotic regulation through things like piezos or things in cell volume changing with development that seem to be really important for establishing both tissue patterning and just maintaining identity. So I think it's really important. And it's something I'm interested in, but be honest, I don't quite have the
So
best way of measuring that. So if anyone does, I'd be really interested in that because it is a question I do get.
Yeah, sure. I got, so the second question is, Rho can also affect shuttling of epigenetic regulators like HDAT, for instance, and that can cause like overall EZH2, etc. That can cause overall changes in in gene expression. I'm not sure if SOX and other genes of interest to you are in that list. I mean, that remains to be seen. So have you sort of tried to look at that angle, look at the condensation state in so that's the question.
Yeah, so we have done working with the HEO lab. What we did was we gave them some cells where we did Rho inhibition and Rho activation, and what we found, if I'm not mistaking this data, it was a while ago, what we found was that with Rho inhibition, we found an increase in clustering with using H2B as a way to sort of measure that using STORM. And then with LPA, it was the opposite. So with the epigenetic marks, I've tried doing Westerns for quite a few of them. And to be honest, I tried, I think 5 or 6 different marks, and none of them ever gave me a consistent change where I would assay for changes in H3K27 methylate or trimethylation that's associated with repression, and then one with an activation. And every time I did it, they sort of averaged out to be basically from, if I compared it to a control, almost directly like comparative to that, where there was like almost no change. But I think we need a little bit more sophisticated technologies to really say whether or not that's happening, or again, be able to collaborate with other individuals that have a lot more knowledge about this than I do from this, my readings.
All right, thanks. Yeah.
Maybe we'll take a question from those in person, and then we'll switch back. We'll go back to the chat. Anybody there have a question in person?
I have one.
Yes.
Ryan, you described that really wonderfully. So
thank you.
So my first question is, Yapintas are homologs, right? Or do they interact with each other?
So I do. Again, Joel's in the room, so they can correct me. But I'm pretty confident that they do interact directly with each other to then go into the nucleus to bind to TEAD. And TEAD is the thing that is actually having that DNA binding capability. And so yeah, that's how they work.
But I guess it wouldn't be unexpected for them to have some redundancy.
Yeah.
And when you knock one down, it doesn't work. So I'm just wondering, is there any situation where you can suppress one or the other and get a big effect, or is it always the case that they express some kind of redundancy in their functions?
Yeah. So the question is whether or not, like, if I can get like where I can orthogonally perturb YAP but not affect anything with TAZ, or TAZ and not affect
not necessarily whether you can. I'm just wondering whether there's situations that have been identified like that.
To be honest, I don't think, looking at a deep molecular, nothing, I'm sure there is something out there in literature. I haven't come across it yet, but I know from listening to Joel Burkle's talks, I believe when they've knocked out singular YAP or TAZ, I believe there's almost like a stepwise effect that happens where knocking out one and then knocking out another and then knocking out both sort of have a stepwise mechanism. But to your question, and I can't recall the exact effects directly. So yeah, to your point, I don't know exactly, but it is something that like that's always in the back of my head though.
And just one other quick thing. That's something you said at the beginning. Actin doesn't go into the nucleus.
Speak up. I'm sorry. Can you speak up, or Ryan maybe repeat what he's saying? We can't hear you that well. Okay.
I'm just pointing out one minor thing about actin in the nucleus. There's a very checkered history of people describing it or denying it. I think there's a general impression nowadays that there's some amounts of actin, not necessarily that would be a cause of your effects on expression, but anyway, it's not completely out of the realm that the actin would be doing something.
Yes, so
not stress fibers, but actin.
Exactly. So there is, so he was pointing out to me that I mentioned earlier that I was saying that none of these really have something like that can go into the nucleus to directly inhibit that. And that was, again, another little misnomer. I meant to say large filamentous actin can, but globular or G-actin can go into the nucleus. And you're right. I think there's a lot of, again, differing results with its effects in the nucleus on various things. Like I've seen just it, and I can't really give an answer as to what it does because it feels like everyone gets a different result with it, where it is. And so that again is something, and I almost wonder, right, with our Rho inhibitor, if we're increasing globular actin, if that could be another way for the cell to sense high or low mechanoactivity that's completely independent of everything. But yeah, that's something that's really good point too.
Good. We'll go back online again. So Ryan, no, I'm reading Joel's and saying Ryan, sorry. Joel, your question, if you have, you want to just go ahead?
Yeah, I'll jump in. So Ryan, I guess the one that I want to talk about, I love that you're thinking about cytoplasmic functions of YAP and TAZ. I think that's really, really important, and I love that you're thinking about it and the way that you're thinking about it. So you did these experiments on soft and stiff to sort of push YAP and TAZ in and out of the nucleus. But do you think that is enough to differentiate between cytoplasmic and nuclear functions? Can you think of a way to really get at whether there's a cytoplasmic function? Because this is something we've struggled with, and I'd love to hear your thoughts.
So the question is, I sort of teased out in the beginning whether or not, YAP and TAZ in the cytoplasm could be directly or indirectly impacting SOX9 versus just going to the nucleus for that repressive role. And it's been an idea where I think there's been some cases noted in the literature of YAP directly interacting with SOX9. This has been mostly described in cancers. Where there's potentially they didn't show this data, but they had a potential hypothesis when I read their discussion that there is a potential binding site for both YAP and TAZ to directly bind SOX9 to inhibit it. That was a little bit of a difficult thing for us because what I would have liked to do, right, is to do a cell fractionation to pull down cytoplasmic versus nuclear YAP-TAZ, as well as SOX9, and perhaps do some immunoprecipitation of that or co-immunoprecipitation of it. The problem is with a lot of our systems, we keep directly impacting total amounts of YAP-TAZ and SOX9. So it kind of felt, at least when Rob and I were talking about it, it felt like whenever we were to do this, it would almost sort of bias something, because it's if we're doing some of these perturbations and we almost completely get rid of that protein, it's almost at least in our opinion, we were oh, I don't know. It's a little bit more of a tricky situation, but yeah, it's something that's really important to us though. I hope that answered the question.
Great.
Yeah, great job.
All right, we'll go back to the room in person. Any questions out there? we'll continue with the chat here. Eileen Shore. Okay.
Hi, Ryan. Really wonderful work. I really enjoyed hearing about it. Just I guess a broader question. You were referring to the chondrocytes and what's happening in articular cartilage, but do you think the same kind of thing is happening in endochondral or cartilage that has an endochondral fate, or something different is going on?
So the question is, like, I guess for some background, right, there's sort of like in development, again, to make it very general, when the limb bud is developing, they'll sort of split off into fates that go into an articular chondrocyte or an or what will become the growth plate or undergo endochondral ossification, to put it very quickly. And they express a very different range of genes, particularly in their matrix. And I believe some of those works that I really hit on from Frank Byers' group, they were more interested in, again, the process of Rho and endochondral ossification. And what they found in their context was using ATDC5 cells and primary chondrocytes was that, I hope I'm getting this correctly, was that when they inhibited Rho, that actually sort of suppressed hypertrophic chondrocytes, or something that would eventually lead to something that, correct me if I'm wrong, Elaine, if I'm using the right terminology, that would lead from a hypertrophic chondrocyte to something like an endochondral chondrocyte. So there seems to be this dichotomy where increased Rho signaling, and from our perspective, increased mechanoactivity would promote more of an endochondral fate, versus inhibiting that would promote a articular chondrocyte fate. It's little nuanced, but with the early work that I didn't mention, it seemed to have made a big difference, where we were able to see those results too, that was sort of opposing ours, but in different media contexts. So when we gave cells something that was just sort of like a basal generic chondrogenic differentiation media, we got where the complete opposite of the results with Rho inhibition, where we actually found a decrease in those genes that I mentioned, SOX9, COL2 aggrecan. But when we put it in a media that our lab commonly uses to differentiate human MSCs or maintains articular chondrocytes in culture, their identity, to maintain that articular chondrocyte fate, that was when we were able to get the results that I showed today and promote that broad range of those developmental relevant genes. And yeah, and I think that showed really strongly with the RNA-seq data with LPA, where most of those processes were all about like endochondral ossification and matrix catabolism. So it does feel like there's like two different sides of it, of like high Rho endochondral, low Rho articular, but I'm not too familiar with the rest of the developmental pathway and how that could interact.
Really interesting. Thank you.
Kyle, I think if there isn't more questions out there in person, then we'll continue here. I think Kyle Vending had a question.
Hi, Ryan. I'm Kyle. I'm a new faculty at Penn Dental and Penn Engineering. Thanks so much for your talk. I was really interested in kind of the implications of your work for using primary cells with materials for regeneration. And so have you thought a little bit about how you might apply these findings? Like, would this also be relevant in trying to differentiate or reprogram MSCs? Or, and if you put the cells in 3D, I mean, can you use what you're finding to also promote chondrogenesis in 3D scaffolds?
Yeah, so I'll answer the first question about 3D culture and whether or not these results sort of would match up with what we would see in a 3D environment. So one thing that I did first to sort of tease at this was I plated these cells in what I call a micromass, which is sort of it's not the perfect 3D environment, but what I do is I will seed a very concentrated amount of cells, and this is sort of, that's been used commonly in this field to mimic limb bud condensation as like an in vitro model to look at that. And when I used, again, these same molecules, CTO4 and LPA. And also I only did this one time, so take it with a grain of salt, where I did do a knockdown of YAP and TAZ in a micromass, and I got almost the same results. The only difference was the magnitude of the increase or decrease was different. And this was just because putting these cells in a very dense environment will really promote, will skyrocket the chondrogenic gene expressions that I mentioned before relative to high density monolayer that I did. And I felt like with CTO4 treatment, what Rob and I sort of talked about was like, I think we're hitting a ceiling on like how much of this expression we can increase further. And so with 3D, it seems like similar trends are happening, but it is an important process to note because others have found, you know, what I showed with YAP dynamics in 2D can almost be the opposite in like 3D scaffolds that you described, where it seems like the stiffer scaffolds, I think this was done by the Sebastian Vega and when he was a postdoc in the Burdick lab, found that as it increased stiffness, YAP actually gets less nuclear with using MSCs. So it's just to make cell signaling more complicated, but yeah, it's not something that we've looked into directly with a 3D scaffold.
Okay. We have another question there, Ryan Locke, in the chat. Is he?
Yeah, I'm still on. I just asked a question if Ryan had seen any or come across any like molecular models of the interactions, protein interactions between YAP-TAZ, TEAD, and SOX9. I think like most of the stuff that I've read on SOX9 and YAP is like with the EMT transition, but I don't know if I've ever seen any modeling of if they can interact with one another. But just curious.
Yeah, so the question is about if there's anything known about a molecular model between YAP-TAZ, TEAD, which is sort of like that big complex that can enact its DNA binding activities, versus SOX9. Some groups, I believe this was Barry Thompson's group, what they found was when they did a YAP knockout model in a mouse, specifically a conditional one with call2, which when you do when people use a call2 Cree mouse model, that's used to come on when those chondrocytes have been specified to be chondrocytes in development, or lymph development anyway. And what they found was that when they did that knockout, chondrogenic factors such as like Alcian blue, or like, I believe if I'm quoting this, or if I'm recalling this correctly, they found basically an increase in chondrogenesis. And when they did ChIP-seq, they found at the very least that that there is a TEAD binding site in the SOX9 promoter that YAP can bind to. And that was sort of, I think, where, if I'm recalling correctly, where it sort of stopped. Others in cancer have found, again, a very direct interaction between YAP binding to SOX9. But in the cases of cancer versus chondrogenesis, where they hypothesize it has a more suppressive effect on SOX9 activity, in cancers, there was a promotive effect where YAP binding to that SOX9 promoter actually increased SOX9 gene expression. And then it did like a negative feedback loop on YAP, where that increase of SOX9 led to an increase of, which signaling, it's always complicated, always ridiculous, but SOX9 led to an increase of a microRNA that then inhibited YAP mRNA. And it was like this very roundabout molecular model. So it seems like this interaction is possible, but it seems incredibly context dependent on both cell type and also point in time of development.
Wonderful, thanks, Ryan. I mean, it's obviously incredibly complicated, and I'm impressed with how you keep track of up, down, and all the different regulations. So great talk, thanks.
I have a very quick questions about the substrate, because in a way, I think we talked about 3D, but again, you go from extremes, glass, which is probably gigapascals, right? Then the 55, and then down to very small. You know, what's your reasoning, I guess, in terms of that choice of that range and staying at the 55?
Yeah, and so The question is, yeah, pointing out that there is, and it's a completely fair too. So glass, again, I think measured is about, like, I agree, is about gigapascals, and we went down a few orders of magnitude to kilopascals. I think the reason why we needed to go so soft was, again, a limitation with our study is we are using a cell line that's been passaged on a stiff like tissue culture plastic for decades, or probably passage 5,000 right now on glass. And there is described by Suchinio and very many other groups where there is like this like mechanical memory. And so our reasoning with this was going down, like we had to really take them down to a very soft substrate to really get them out of that I'm on a stiff, very super physiologic environment. And that was sort of how we reasoned that. And other past individuals in the lab finding that these were like 55 and 5 were like the two ranges that we sort of term, even though they're really kind of comparative terms. Soft stiff was 55 and 5 to sort of be like 5 kPa, 5 kilopascal. Gels are very soft, more physiologic, with getting that cell roundness and native chondrocyte like state, and 55 was our intermediate. So it's a definite limitation though of the study.
No, that's good. I mean, I think it's always optimizing and figuring out where to be on that. So it's very good. Yeah. Okay. Any other questions? All right. Well, thank you, Ryan. Let's thank the speaker for very exciting talk and very exciting work, finishing up your dissertation work.
Thank you.